Good morning, everyone. I'm Simran Da Costa, and on behalf of Alton Technologies, I'd like to welcome you all for another very informative virtual session. Our session for today is entitled Role of Chemo-Bioinformatics in Natural Products Derived Molecular Medicine. Now, before we begin, a small introduction to chemo-bioinformatic tools. We've seen in our previous sessions that there are a lot of natural products that are used in medicine, and we've also seen how bioinformatics tools aid in this process. Now, chemo-bioinformatics tools, they can enhance the efficiency of natural product-based drug discovery and development by leveraging computational approaches to analyze models and predict their biological activities and therapeutic potentials. So, by applying data mining techniques, chemobioinformatics extracts valuable patterns and relationships from large data sets of natural product information. We see that computational tools can identify new therapeutic applications for natural products by repurposing them for different diseases or optimizing their chemical structures to improve the efficacy and safety of profiles. And these techniques enable virtual screening of vast libraries of natural products against specific biological targets. We also see molecular docking simulations predict how natural product molecules interact with target proteins, and this helps us identify potential lead compounds quickly and cost effectively. Moreover, we can also explore the structure activity relationships in natural products. So, by correlating the chemical structures of compounds with their biological activities, researchers can prioritize molecules with the most promising pharmacological profiles for further investigation. And this facilitates the discovery of novel drug candidates and biological mechanisms. So let's understand the perspectives about chemobioinformatics in molecular biology from our expert. It gives me immense pleasure to welcome Dr. Gopimohan. So Dr. Gopimohan is a professor at Amrita School for Nanosciences and Molecular Medicine, Amrita Vishwa Vidyapeetham Kochi. He has received several awards and fellowships. He was cited as an international well known expert in the field of structural bioinformatics and chemoinformatics by Synergics Limited, United Kingdom. He has received Medical Research Council Fellowship, UK, CNRS Associate Research Fellowship, France, DPT Travel Grant Award for Invited Talk in US Conference, Dr. D. M. Bose Best Poster awarded on the homology modeling studies on human alpha 2C adrenoreceptor protein. He was awarded ICMR Senior Biomedical Scientist International Fellowship and visited Computational Biology Lab of Professor Jerzy Rudzinski. He has professional societies membership at American Chemical Society USA, life member of Indian Society of Chemists and Biologists India, member International Society for Computational Biology, Virginia, USA. He was the principal investigator of DBT ICMR projects titled Structural Bioinformatics Analysis for Glia Maturation Factor Beta, target to exploit its immunomodulatory role for the treatment of multiple sclerosis disease, integration of in silico and bioassay techniques for the identification of novel inhibitors against mycobacterial LD transpeptidases, clinical translation and new frontiers of medical nanosystems, development of PD1/PDL1 checkpoint inhibitors for adjuvant therapy against sepsis. He has guided several MTech, MSc, and PhD students and has supported them during their projects and research work. And he has a total of 95 publications. So, without taking much time, I'd like to hand over the platform to our expert. So, welcome once again, sir, and I invite you to take over the session.
Okay, thank you, Simran, for the introduction. I think you can hear me. I
Yes,
think,
sir, we can hear you. Yes, sir.
okay. Thank you for the introduction. And I think I have to open my slides. Okay. So, thank you for inviting me to this webinar also. Yeah, Altum Technologies, we all use Discovery Studio software for our computational drug discovery program. But today I will focus on the role of the chemo-bioinformatic techniques in natural products derived molecular medicine, because this is a very vast field, natural products as itself. And we are already this in ancient or Ayurveda system of medicines, which generally deals with herbal medicines and many Ayurvedic ingredients, which we call it as broadly as a polypharmacology. So some of the aspects we can touch upon using chemo and bioinformatic techniques, which will be covered in this natural product derived therapeutics, which is a very important theme for this webinar, which Altam has initiated, and I really appreciate this initiation. And today, my talk will be the first part will be the general introduction of the molecular medicine, how the molecules will be involved in uh, molecular level mechanisms of protein or structural bioinformatics, as well as the chemoinformatics, how it can be efficiently utilized in integrating with the wet lab um, and animal model systems. And the second part of the talk will be the natural product, how the molecules will be utilized, and what are the different techniques which one can use for this computational aspects. And the final part will be two different case studies in which we successfully identified natural product derived molecules for TB as well as for multiple sclerosis. So that will be just shown as an example to show how computational biology can be exploited to use efficiently in uh, in natural product derived medicines. Okay, so before that, I just want to show you this common slide actually, in which I have shown this is a Linus Pauling. Actually, you can see his picture here, and he had delivered a Nobel lecture in 1954. It's very more than 60 or 70 years old. So at that time, he told that we may, I believe, anticipate that the future of the molecular medicines will be the structure of proteins, nucleic acids, polysaccharides, and other key substance with higher molecular weights, and it will come to rely upon new principles in chemistry involving precise geometrical relationships among the atoms in the molecules. So this was he anticipated in 1954, but at present we already experiencing all these things using all our data sets in our hand. As an example, we have the Protein Data Bank (PDB), which all of you may be knowing, as well as Cambridge Crystallography Database, which deals with the small molecules, in which the crystal structure of all these precise geometrical relationship of these macromolecules such as proteins, nucleic acids, as well as small molecules are available in all these different databases. And in at present, we will call all this as a big data sets. And how he has further suggested that how we can rigorous application of the new structural principles. So we are all adopting now using chemo-bio as well as many other quantum mechanical structural principles, and that great progress will be made through this technique in the attack by chemical methods on the problems of biology and medicines. So this was a classic talk or the statement which he has delivered in the Nobel lecture in 1954, and after reading this, I really want to put in front of you that we are really experiencing using the data we have at present, and how we are exploiting using nowadays using the very terminology like machine learning, artificial intelligence, as well as implying using all this big data which are already available with respect to our proteins as well as small molecules, including natural products. So this can be easily implemented in the natural product derived molecular medicine also, which we call in the Indian system of medicines as Ayurvedic medicines, which also have active ingredients as a natural product, which do play a very important role in the therapeutics for different diseases, which we will be seeing in or which we already have being developed and discovered in various types of diseases and which are also well known. So, this slide also have the same thing like what Linus Pauling has dreamed towards modern medicine can be transformed to Ayurvedic medicines. So that should be the question now. It is with respect to our latest technological development. Can we transform this to our natural product derived medicines or the Ayurvedic medicines? Because there are differences between the modern medicines as well as natural product or Ayurvedic medicines, which I think all of us know actually, because natural product derived medicines do play an important role, but uh, which is a, uh, which is in culmination with Ayurvedic medicines play a polypharmacological role in which multiple molecules or uh, different varieties of natural products in combination, synergistic or combination or additive manner will play an important role in curing the disease. So it is uh, completely different from the modern medicine which we generally deal with, in which we will generally talk about single molecules and its pharmacokinetic and dynamic properties. Of course, it may be now changed to systems biological approach in which they will deal with multiple targets uh, for these modern medicines. So we have big data in natural product derived medicines or Ayurvedic medicines, which why we now also coined a term called Informatics. So this is also well-known terminology nowadays, which we can tell or which we can designate as natural product derived molecules, or the molecules mainly dominate with respect to the naturally occurring molecules. So bringing your data to life. So how we can bring this all the important information of this data or the molecular aspects to the really curing the disease or the life process actually. So the latest trend in applying this, we call it as all machine learning and artificial intelligence, along with all the methodology and the techniques which we implement in solving these problems. So the main interest, or the main concern or the understanding is the mechanism of action of these natural products or the Ayurvedic drugs, which is very very challenging actually at the molecular level, which the Linus Pauling has earlier depicted that the attack of the problem at the molecular level, how we can, of course we have the structure of many molecules now in our hand, which is, as I mentioned earlier, in Cambridge crystallography database or in many other different databases including protein data bank, all the molecules. So can we be able to utilize all this and understand the molecular mechanism of action of drugs, of at least all the FDA approved drugs, which we have at present. Hardly we had around nearly 1,500 to 2,000 FDA approved drugs. Can we be able to know the proper mechanism of action of all the FDA approved drugs? Still, we don't know clear cut molecular mechanism of many of these FDA approved drugs. Even our technology has developed so far. So the formulations at the molecular level is also another challenging things, because it involves multiple kinds of molecules which are in the polymeric form or in the different formulations, and it is also a challenge for the chemo or bioinformatics technology, or how these techniques can be implemented in the Ayurvedic system of medicines. So before going to the talk, I just want to show, as I mentioned, the big data actually how from how, where it is coming, and nowadays we have the tendency to call artificial intelligence and digital era towards natural products and informatics. So the words look very attractive, but we have to conquer, or we have to understand better by taking or by making use of all this big data available, or the making use smartly all the data available in our hand in different databases at least, or we have to at least put all this in one place. In Indian system of medicines, we have the Ayush research portal, which I think some of you will be knowing actually, and they have developed different databases and different um, uh, data to put together, which include digital helpline for Ayurveda research articles, which in short form called Dhara actually, and Ayush research portal, and the tradition knowledge digital library, which is called TKDL, or Rudra, which is random uninterrupted documentation for retrospective analysis program. And different software like IU software also developed using CDAC, and Prakriti Vichaya and Trishanga Kosha. So all these things and many other different softwares in the natural product derived or Ayurvedic application towards different therapeutics have already been developed and being utilized using all this big data available, and strengthen the position of Ayurveda in the digital era is the main theme, because India, like China, we also use Ayurvedic drugs for different diseases. So we need to understand, at least using this big data, what are the different molecular mechanism of interactions or molecular mechanism of action, at least using all the techniques or tools available in the chemo or the bioinformatics field, or we have to develop new techniques in this area. So traditional knowledge digital library, which I mentioned earlier, so this has around 1,200 Ayurvedic, Unani, and Siddha formulations. So this is also our ancient system of medicines, which have a lot of information in different books or different ancient books. So also have it as 4.54 lakh medicinal formulation is available to users, and patent office under TKDL SS agreement, and it also have the tool to understand the codified knowledge existing for the Indian system of medicine, including different system Siddha, Unani, and Yoga as prayer art. And it is not a diagnostic or usage database, as all of us know. It is only for the information sake, but the information is well assessed actually, and properly. And this is a collaborative already project with the CSAR New Delhi, as well as the Department of Ayurveda and Yoga and Naturopathy, Unani Siddha, and homeopathy, which we call in short form as Ayush. So all these are the big data which we can utilize in our latest trend, like machine learning or AI kind of applications in the natural product derived medicines or in the Ayurvedic system of medicines. So all this can be utilized. So that's why I put up this slide also here. And there are supernatural to database, which is also available, uh, which is being developed from a German university actually. Supernatural to provides a highly curated online database of natural products. I think some of you will be knowing actually, and this has many thousands of natural products extracted from various sources actually. So this offers an additional search and analysis option. So it also has some data mining approach, and you can also predict ADMET or PKPD kind of toxicity prediction for the database compounds, natural product derived compounds. Also, they have built a very nice website and published a nucleic acid research kind of journal actually. So, for instance, a substructure search can be performed to identify compounds containing this substructure actually from the supernatural to database, and additionally possible target proteins can also be find out mechanistic signaling pathways can also be predicted for these particular natural compounds based on 2D structural similarity search to known drug molecules. So, so if someone want, this is the, I think, the website which is available actually, and there are two different papers which is already be published in Nucleic Acid Research, which can be downloadable. It is open access, and you can read more better actually. So this is the latest of this particular database, and this is Robert Prisoner. It is developed in this professor lab um, in University of Berlin University in Germany, and one Indian author also here. You can see Priyanka Banerjee, and so they collaboratively developed this supernatural 3 database. I think some of you may be knowing in the audience natural products and natural product based derivatives. So these are the rich informations, as I told you, big data, or in our uh, in our platform, which we can easily exploit for our natural product drug discovery. And recently, you can see in the Nature Review Drug Discovery, there is another classic paper which came around, which we call as Artificial Intelligence for Natural Product Drug Discovery. So these are all the very high impact journals. The impact factor is around 120 for this review article. So these are the high-end articles which can be utilized to get information of the latest trend, and how we can utilize all this data by discovering from this literature in our data mining, as well as our drug discovery program in the natural products and derived for different therapeutics. So, so this is a supernatural 3 database outlook actually. So here you can see the different um, applications of this one, and there are some examples with respect to COVID-19 as well as other pathways. So there are various functionalities available in this, including the Chembel database they have integrated, and you can also get the mechanism of actions and the disease indications, as well as it is also integrated with the different systems biological pathway, like human pathways and different pathway analysis, which is cake pathway kind of thing, different pathway which proteins are interacting in the signaling downstream signaling mechanism of the particular compound or the particular protein of interest with respect to the particular natural product. So all these things can be made utilize, including some of the organoleptic, which include like sour, sweet, or taste of the particular compounds with respect to, so all these things, some properties including ADME, PKPD properties, and even one can focus target libraries also. So all these are contained in Supernatural 3 database. So it is very important database in the natural products. So that's why I just shown you this one. With respect to small compounds, natural product derived compounds, and this is also another version of this one. You can build new molecules in this, like zinc database. I think some of you know here, where we can put the smiles notation of the compound and get the structure actually. So here also you can put the smileys and get the structure of the molecules and calculate its properties, and also its functionalities, etc. So there are various options available in this website, which practically one can perform, and you can design new molecules also and predict properties, or do Tanimoto coefficient derived structural similarity search, and get mind like 90%, 80% kind of structures you can retrieve from the databases, and you can quickly assess the similar drug like molecules, which can be exploited further for your in silico drug discovery program. So further adding to the same thing, we have the some CDs or NLM, National Library of Ayurveda Medicines, also have some botanical information. So this also a dhara like that, and more than 8,000 articles has been put into this particular website, and one can also get in the CD or in the pen drive nowadays, or so that you can access this old or the comprehensive data in the plants derived Ayurvedic research, which also have direct link with the natural product derived medicines. Yeah, and so one can exploit this information also for your analysis. So this slide I just put you actually just to give you an information like Nobel Prize in chemistry like 2009 and 13, and 2013 was mainly meant for the structure function relationship of macromolecules as well as the computational modeling. So that is the main thing actually. Here there is overlapping of the slides, I am sorry, but here the three scientists like Ramakrishnan, Keats and Yonath got the 2009 prize for getting the structure of different ribosomal crystallography. So that is protein structures in the ribosomal field. They have proteins they have solved actually, and which is a direct relationship with the antibiotics which we can consume like streptomycin or different antibiotics, which will make this or block this synthesis of these ribosomal proteins, so that further infection can be controlled. So they have done a lot of crystallography and cryocrystallography work in the structure-based drug design towards ribosomal proteins. So their pioneering work has given them the Nobel Prize in Chemistry in 2009. And further, the 2013 is Martin Karplus and Aaron Welch and another professor also is there. So they shared the Nobel Prize in 2013 on the basis of development of multi-scale canonical system, which we generally use in our Discovery Studio, the charm-based force fields and other multi-scale modeling system, which is already implemented in different modules, I think, in the Discovery Studio programs. And Martin Karplus and their force fields and other theoretical analysis do play an important role in our molecular modeling studies as well as structure-based drug design. So modelers react to the chemistry award, or we are good at guiding experimentalists. So that is the main thing actually, which is published in Nature actually. So I just want to emphasize the importance of the in silico or the computational chemo or the bioinformatics, how it is being integrated with the in vitro or animal model systems, and how it will be utilized in guiding different experimentalists, so that they can utilize efficiently the in silico derived molecules or the natural products for the future biochemical or animal model experiments. So prices prove that theorists can measure up to experimenters. Generally, the theoreticians are considered to be in a different world actually, and may not be very highly integrated with the experimental methodology or the techniques. But this award and our own field will will generally nowadays being successfully integrated in various applications, including different diseases, and we can clearly suggest in silico or the computational biology do play an initial guidance or the in the preclinical drug discovery programs. And as I told, like computers and biology have not been sufficiently appreciated. So that's what I meant. It is not sufficiently appreciated earlier, but in the coming era, since we have the big data and machine learning and AI, artificial intelligence do play an important role. So this they started dominating now all the experimental and our livelihood. So they clearly will dominate in the coming 10 years or so, and guide the experimentalist what type of biochemical experiments they can plan, and what kind of without killing of animals they can safely test different chemicals by in the animal models also, without killing them. So earlier we do not have the toxicity predictions. Now we have ADMET and many other different softwares which can be able to predict PKPD as well as the toxicity profile of different compounds. So that is very useful. Actually, in that field also we have grown very nicely, and that will also be very important for the animal model experiments. So without doing much harm to the animals, we can predict different type of molecules for animal model validation. So some of this I may also show you in the since the time is limited. And artemisinin, as you told, this is also one of the natural product which all of you know. Artemisinin is a wonder drug, phyto drug against malarial parasite, and the half of the price was shared by this this Chinese scientist as well as from Campbell and one Japanese scientist, Umura. And this is derived from the planet Mysia, actually. And the structure, you can see the natural product structure. Actually, we have the structure, and it is in the Cambridge structural database also. And we have the clear-cut analysis of what type of atoms are present in this particular active ingredients or the natural products, and which will be really useful in killing the parasite. So we have all this knowledge actually, and informatics revolution do play a very important role nowadays, and it is alive and kicking, as I mentioned. Now, with the help of machine learning and artificial intelligence, we are now psychologically also transforming all the things. Initially, now we will do some of the theoretical stuff before jumping into wet lab experiments or some medicinal or pharmaceutical chemistry work. So unless you validate in silico or computationally, we are psychologically not properly done actually. So all this, even IR informatics is growing very well. So that's why all this workshop webinar is coming up nowadays, and we have to develop more skills in this area also, because this is our ancient system of medicines. So actually, fast forward in 2030, actually, how we will see actually after 5 or 10 years, what to expect in molecular medicine or the natural product derived medicines, and we will call also nano medicines. So all these are the different terminology or the different field of interest which is growing exponentially, and which is growing very fast. And in all these fields, informatics and the computational biology do play a very important role. So in this slide I mentioned, like, Ayurvedic doctors, can they use smart card actually? So some of this thing has to be developed, and I think few of the modern medicines nowadays started using smart card, and we all have all these things. And it should start with the natural product derived, or Ayurvedic doctors should also be able to go into this digital era and use this whatever the Ayurvedic formulation, everything should be digitalized and put it in, so that the patient's level smart card can be developed, and it can, and the doctors can do more personalized medicines in this area also. And the nano medicines, this is another field of interest where nano drug delivery and other things are playing an important role. So I'm not going into details of this one. So smart card in Ayurvedic and traditional medicine has to come in this modern digital era, and one is expecting the personalized level medicine also, and all of our genome should be get sequence within thousands of rupees. It will be in the coming era, and so all these things we are expecting in the coming 5 to 10 years, so that one can have some personalized derived medicine rather than general medicine which we are nowadays using. And the trend should change actually. So the omics revolution, as I mentioned earlier, so there are different terminology which we have coined, like pharmacoinformatics, which we have mentioned here. The term pharmacoinformatics, it was initially coined in when I was in NIPER, actually, in the National Institute of Pharmaceutical Education and Research. There we called. So this is nothing but an integration of the bioinformatics and the chemoinformatics. And there is a specific department in all the NIPERs, and maybe nowadays it started in different universities also, where they build the role of informatics revolution in the drug industries and the drug discovery programs. So in the drug discovery, so bioinformatics can also be integrated into this pharmacoinformatics, and which will do play an important role in the natural product derived medicines. So all these things has to come up in our academic sector as well as in our industry sector in a more better way, and so that one can guide actually better in the drug discovery program. So in this slide I just want to further utilize the strategies. What are the different strategies which one can utilize in the modern as well as natural product molecular modeling? Or so one can be drug based or the natural product drug based, which we call it as it has nothing to do with the target, but it is only using the drug derived small molecules. And another one is the structure based, where we will take into account the protein target also into account in the molecular modeling actually. So the natural product derived have structure activity relationships, which can be either 2D, 3D, or 4D, and different dimensional QSAR, that is called the quantitative structure activity relationships. So this is also a well developed field actually, but in the natural product it has yet to be developed properly, and we need to identify proper molecular descriptors in this area to understand the proper structure activity relationships. The main problem of natural product is its stereoisomeric form, RNS form, and many other different chiral compounds, which I will be showing in the coming slides. And so, and getting a proper confirmation, will say the bioactive confirmation of natural product is also a challenge actually, and how it is interacting with the proteins or enzymes is the next challenge actually. So this one can develop further actually, and to identify the lead identification, and one can also predict in the natural product how we can develop a good in silico PKPD or ADMET kind of predictions, and one can predict the better lead optimization in this particular field also. So the supernatural database of natural products, which I have shown you earlier, can be utilized for doing all these kinds of studies in which we know the structure of different natural products. So another one, as I mentioned, the structure base actually, which include the crystal structure analysis and the homology modeling, which also we generally do, because we have also now AI based modeling of the proteins, many different servers. So almost we have solved many proteins, and its models are available actually. So this structure base is also very utilized, can be utilized in the protein natural product interactions, and how this different natural product is interacting with the active sites of the protein is also important actually. And one can even modify the natural product by introducing different fragments, functional groups, and to understand its better binding affinity, which one can perform using the molecular docking technique to understand the docking score, or the, you can say, the binding affinity. So all these things can be understood actually, and but the challenge is next to synthesize those fragment based modified natural products. And one can also build a pharmacophore model of for different natural product drugs, and few papers are also available. How this pharmacophore model can later on utilize in virtual screening. Virtually means you can, like maybe you can utilize different natural product derived databases for virtual screening, database screening, so that one can prioritize different hits or different compounds in the preclinical prioritization of the hits after performing the EDMET analysis. So this is drug based as well as the structure based. One can have two different strategies one can adopt on the case by case. You can adopt uh, in in all these different cases. So further coming to this one, like Sightline annual review, this publishes the latest trend actually in the drug discovery program and the R&D pipeline size by years. Here you can see the drug count actually left hand side. We have, it is progressing well. You can see the curve is increasing actually nicely by the year actually 2023. If you see in this, we have around new molecules, 21,292 new molecules, which we have discovered actually. 2022 we have 20,309. So we have R&D pipeline size by year. Preclinically we are very good actually. We have around 21,000 new compounds in the preclinical era. But here in the right hand side here you can see the sharp fall actually once you go to the clinical trial, actually how this will culminate, and further you can get hardly launched around 1,000 few molecules also, and this is also very many years of work actually. So in the pipeline of developmental phase, actually here you can see stages of development and the drug count actually is from 12,000 or something, how it is sharply decreasing, and toward the launching after all the clinical trial studies, ratio is reduced and meaning gets suspended also. So what are the reason of all these things, and how we can reduce this downgrade graph, and you can get at least some better graph. So what are the technology, and how the chemo and bioinformatics can be able to help the pharmacologists as well as the medicinal chemists to develop better drugs with less adverse effects. So can our big data, or especially in the natural product, can also guide them, and can reduce this barrier actually. So this is a model to increase efficacy and reduce toxicity. So this is our challenge actually in the particular modern medicine, as well as the natural product derived medicine, how to increase the efficacy as well as reduce toxicity actually. So there are various natural product, and there are different things which one can perform. The topics of interest can be like Ayurvedic derived natural product designing, or the one can use the machine learning also, which I am not going to details. And there are large language models like LLM, which is also nowadays very popular. So can Ayurvedic, even Sanskrit and other languages, computational languages in Informatics can be developed, and some of them are even developed actually, and distributed electronic medical records in Ayurveda or the natural product can be also being implemented. Medicine plant database for Ayurvedic formulation can also be of interest, and genomics and network pharmacology can also be integrated. So this some of these things we have seen in the supernatural database and application of artificial intelligence in Informatics. So these are some of the latest, as well as integrating with the machine learning and artificial intelligence do play a very important role nowadays, because it is highly automated nowadays. So one has to look upon all these topics which can be um, taken. So the challenges behind all these things, as we are discussing like that, so we call it as a bioactive or the active ingredients or the natural sources. So the computational approaches do have different challenges for tackling these molecules. Like were often viewed as too complex. These molecules are often viewed as too complex. Why it is so complex? But then have been recognized as a valuable source of interesting and diverse structures. Since we cannot leave this is gift of God or something which is provided from the nature actually. So we have to deal and using our different technologies, and what are the things like natural products show higher molecular weight than synthetic drugs, because these are high, and it will not follow the Lipinski rule of 5, which all of you know. I think like molecular weight should not be a 500, should not be more than 500. Most of the natural product have the molecular weight more than 500, as you know, and it contains more oxygen atoms, fewer nitrogen atoms, 3 times more stereo centers. So this is another challenge actually with respect to natural products. Many different stereo centers are the degree of unsaturation is higher. Degree of unsaturation of this particular natural products is higher than modern medicine, synthetic drugs, and they incorporate less aromatic rings. So these are the things difference between the modern medicine as well as the natural product derived medicines, which we have to tackle properly. Although natural product in general contain more rings than drugs, most of them are non-aromatic and part of single fuse ring systems. So these are the challenges actually to synthesize the derivatives of the natural product. Is very very challenging, and is time consuming and very costly also. So that's why our structure activity relationship or fragment based drug discovery is not progressing well due to its high cost and less viability and many different challenges. So like this, the structure here you can see many different chiral centers. Here you can see the dotted one, as well as donor and many different molecules. You can get actually bioactive compounds from natural sources. So this, even optimizing computationally all these molecules look very challenging, and even different isomeric form, R and S form, have different activities. So that is the main challenge actually. Different isomeric form have different biological activities, and how to deal with all this, and synthesizing particular compound itself is a very big challenge actually. So, computational challenges in natural product or Ayurvedic drug discovery include increased molecular weight and a higher number of saturated bonds, resulting in higher molecular flexibility of Ayurvedic active ingredients. And as a rule of thumb, docking and conformational sampling becomes slow and inaccurate when a molecule has more than 7 rotatable bonds. So it is very challenging because of its multiple conformations, and it may take longer computation time. Even one can perform the virtual screening properly because it has a large number of different conformations and different stereoisomeric forms, and too high degree of flexibility may also result from skid that is fitted into enzyme protein binding site, which is also highly flexible actually, which is not properly explained the molecular mechanism of interaction, which stresses the need for visual inspection of virtual leads and selection of the most possible virtual leads. So all these things are the challenges actually. Another putative practice is a higher number of stereoisomers, as I told you, are often during database, and therefore the programs stereo which unnecessarily increase the complexity of the particular problem actually, or the computation techniques. So these are the challenges actually for the particular things, but still one can derive this workflow like this one, like three-dimensional natural product database. Maybe you can have the virtual screening of these virtual hits actually, and available natural product for the biological testings by after doing the selection criteria ADMET. And as I mentioned earlier, those strategies can be applied with respect to natural product derived, like anti-targets, off targets, additional beneficial targets, and all these things can be also applied actually. And further, you can also promising natural starting material. So, what is the starting material after these thousands of molecules can be screened, and what are the starting or the best available virtual screen compound after docking ADME, and all these things can be further used for experimental validation analytics, bioanalytics, and other in vitro analysis from the testing of different extract, plant extracts, and other things, and also can be bioassay guided isolation can also be done. Parallelly, we can also perform some virtual leads and the biological evaluation of all this in order to further culminate to the bioactive natural compounds. So these are the it's not a very straightforward technology, but here the computational effort can be put actually to reduce the experimental effort, and which will also reduce the time as well as the money actually. So both the time and money can be saved actually by integrating this workflow actually using in order to get the natural product derived drug discovery. So this is also another way of putting it, like just screening on respective targets, target based like the protein base. Also, one can extract the screening actually active natural starting material, empirical knowledge or phenomenal effect. So this is also nowadays being implemented in machine learning or artificial intelligence nowadays being heavily used. Actually, so all this technology, multi-dimensional technology, can be bring into actually to understand better to how one can better come up with the starting material for isolation or identification, or the phytochemical analysis of the particular extract can be utilized in order to understand which are the active ingredients present in the particular plants or herbs, so that one can utilize or efficiently use those active ingredients or the natural products for further biotherapeutics. So this strategy can also be applied actually, and further can be validated using animal models or protein-based, target-based. Also, one can do biological evolution. So the computational workflow for the integration in the natural product can be applied using this workflow also. So another case study which I just want to show you here is the EMTB, like Mycobacterium tuberculosis, in which we applied virtual screening with respect to, or you can say the drug repurposing techniques, in which we identified a new natural product in the TB, curing TB, because TB, as you know, is very pathogenic bacteria, and it's a TB regimen which have many different multiple drugs has to be taken for 3 to 6 months. So it's not a very straightforward, and it is very dangerous, and it is how to control the TB is a very challenging task actually. And Robert Koch in 1882 found out this particular organisms. and so this is highly contagious and spreads through air also. So it's very dangerous. And so how one can control, because nowadays antimicrobial resistance are playing a very crucial. So one has to discover new drugs in this area also, because especially in Western world it is not there, but in the tropical countries, in India as well as in South Africa and other tropical countries, TB is still a deadly disease actually, and many different resistance has been developed actually. So it is very important actually. Here you can see many million deaths actually, more than 500, and this in Indian also around 2.7 million cases are there actually globally if you see. But in other places it looks almost okay, but we also still have a lot of burden actually for this disease. So because we generally have the standard treatment like 2 months and 4 months for this particular FDA approved drugs, rifampicin, isoniazid. So can we modify this particular drug, or can we add some adjuvant or some particular drug? So that is our idea actually, or our hypothesis. Can we modify this regimen to treat the TB disease? So can we attack some of the important druggable target, so that we can kill this particular pathogen, so that one can have some control over these particular drugs. So, because of large drug-resistant tuberculosis (MDR) and XDR, MDR TB cases, so multiple drug-resistant TB cases are also very thing in India as well as in different countries. So, so there are different um, papers being published actually, like meropenem clavulinate is an effective, and so these are the already effective antibiotics and the drugs which are available to drug-resistant mycobacterial tuberculosis, and we have this injection actually of meropenem, and limitations of carbapenems are their poor oral bioavailability, and it has to be injected actually intravenous. So it's not patient friendly actually, and we have to hospitalize a particular patient, and it is treatment cost is also very high. So can we modify this particular this combination, or can we give some oral drugs? So that is our challenge actually, or that is our research hypothesis. Can we give some oral drugs so that one can reduce? So in order to tackle this, we identify a new druggable target in TB, which is called LD transpeptidase. So this is involved in peptidoglycan actually synthesis actually in the mycobacteria. So this particular protein target is not available in eukaryotes, so it's safe for humans. So it is specifically there in the prokaryotes as well as in some pathogens. So we generally take, like all of you know, amoxicillin, which will target is a beta lactam oral drug actually, which will target the DD transpeptidase enzyme. It's a classical as well as, so our main aim is to target some non-classical LD transpeptidase enzymes. So how to develop? So on this mission, we try to understand what is the molecular mechanism of action of LD transpeptidase inhibition, which will really will be able to kill by cell wall lysis, and bacterial death can happen. So in this slide, you can see the amoxicillin, and further amoxicillin clavulanic acid will be taken to kill the particular bacteria, but it will be like beta lactamase enzyme degrade the beta lactam antibiotics. So this is amoxicillin function. So still it will be more better actually, but still here you can survive. The bacteria will survive by non-classical PG cross linkage. So can we target this non-classical? So that is our idea, which include LD transpeptidase enzymes. So we have targeted these particular enzymes in our studies and identified a new natural product. So LDT MT1 and MT2 is well known actually, in particular two functional LDTs. So here we got the DBT project also funding for this one, and we have the crystal structure also for this one. So can we identify the active sites and do some structure-based drug design and computational repurposing? So we already know the active sites of these particular proteins, and it is well conserved for this particular enzyme. So can we use this for our TB drug discovery program? So the objectives of the study is the identification of an inhibitor targeting LDT enzymes of MTB and validation of its antimycobacterial activity in combination with amoxicillin and clavulanic acid. So the main part is the computational drug repurposing of FDA approved drugs against this particular LDT enzyme. So that is our first aim and objectives, and further in vitro and in vivo validation. So I will be just showing the in vitro and then showing you the another case study actually. So here our main objective is to do some virtual screening of some FDA approved drugs actually, because we thought it will be more easy to get the already repurposing that will also serve actually to in the drug discovery program. It will be much easier than finding out some new chemical agent and going for regulatory purpose. So drug repurposing, as all of you know, actually this is reprofiling of known FDA products for new therapy actually. So already its PKPD as well as clinical trials, everything has undergone very nicely in regulatory aspects. Everything was successfully performed. So we have all the data in our hand. It is only to check whether this particular drug is utilized in some other disease conditions, some new indication. So a lot of drugs has been repurposed in this slide, which is shown here actually. In this original indication like fungal infection can be utilized in leishmaniasis, or in cancer methotrexate can be utilized in cancer, can be utilized in psoriasis and rheumatoid arthritis. So like this way we also thought can be repurposed in our and can be used those in the TB cases. So here we have repurposed actually here in this slide using FDA approved drugs and with respect to LDT, MT1 and MT2 enzymes. And here we identified using, we have done molecular docking of around 1556 FDA approved drugs, and finally we could be able to get high docking score compounds, more than 8 actually, with respect to MT1 and MT2. And I'm not going into details actually, but this here finally we got the overlapping hits. Overlapping means which is more common to both these particular enzymes, MT1 and MT2. So acarbose and diosmin actually. So here you can see both of these are naturally derived compounds coming from plants or the fruits actually. So one can classify this as naturally derived compounds actually, and here we got very good docking scores. And we have considered diosmin because of its better bioavailability and better ADME profile than acarbose, which has some bioavailability problem actually. So can diosmin be a repurposed drug, can be utilized in TB purpose along with the TB regimen, which I have shown you earlier, like amoxicillin and many other drugs, which we can be introduced diosmin as an additional drug, or as an adjuvant for a TB patient. So with this mind, we have further gone, as in this slide. Just I want to show you like the diosmin. It is a flavone derivative, and one can get this particular compound from this. Um, it is a nutrition supplement actually, and in this lemon etc and citrus family you can get this one, and here in the market also it is available in this particular thing diosmin. And so it is an antioxidant, anti-carcinogenic, anti-inflammatory properties. So these are well characterized and it is well known actually, and it is a nutrition supplement. So can we just give this diosmin to TB patient just to get at least if you can reduce the time of the medication, that itself is a very big thing actually. So we have performed like molecular docking of this one with respect to LDT, MT1 and MT2 with respect to diosmin, also shown here actually, and particular things, and further molecular interaction analysis, and further we have done this molecular with respect to particular important residues. I think I am running out of time, but all these things, iron bonding interaction, all these things we have studied with respect to this particular natural product and how it is interacting with the active site residues. And further, we have the knowledge of all these are the active site residues of these particular enzymes. And this is a bacterial growth index, actually MGIT, bacterial microbacterial growth index tube, actually 960 system. Here you can see the AMC, amoxicillin clavinic acid, do not show much inhibition for the bacterial growth. But when we add diosmin, DIA is diosmin, AMC plus diosmin, automatically this growth index has reduced and was studied. And MTR resistant indium growth at 20 microgram per ml of AMC in combination shown growth has been reduced and it has become better actually. So in this case also here you can see the anti-tubercular activity of AMC divestment towards MDR TB also we have checked actually, because here we have the Amrita hospital in our hand. So there are many clinical isolate also can be tested actually. So we have tested using this bacterial growth index MGIT tube actually. And here also when we add diosmin actually, here you can see there is suppression of the complete growth of the MTB actually when we add this much 500 microgram per ml of diosmin into this particular AMC. Without adding, you can see how this graph is, the trend is actually. So as you know, the MDR strains of the first line drugs, all these drugs are not at all good. All these have the resistance and some are sensitive actually. So all these things lead us to identify diosmin can be utilized as a add-on therapeutics, and published in scientific reports combination of repurposed drug diosmin with AMC cause synergistic inhibition of the mycobacterial growth. Okay, so this is a conclusion actually using computer-aided drug repurposing or via infrared diosmin, and further in the MDR MTB clinical isolate also we have seen, and this we have tested in M. marinum because MTB is very dangerous, or in this particular lab setup is more. So we have tested in the Melanogaster TB model, and further we published this work actually. Okay, so the second case study which I just want to highlight is in the neuroinflammation, that is in multiple sclerosis. So this is in collaboration with Krishna Menon, which is a proteomics and biotechnologist. And so here we have also done some natural product derived database derived compounds. Here we have initially homology model a important neurological target, which we call it as GMF, glia maturation factor actually. And homology model of this particular neurological target has been identified, and we have predicted the active site like site 1, site 2, all these things we have predicted actually, and we have done sequential virtual screening of some natural product database and identified few of the hits. Actually, there are many drugs which are coming with respect to structure-based drug design. So this is a workflow actually with respect to specs database. As some of you may be knowing, all these are the naturally occurring compounds. So we took around 9,61,000 molecules actually, and did the virtual screening of this particular thing and using docking as well as leak filter actually with respect to that. And further finally four molecules were retrieved for in vitro testing. So this is a power of virtual screening from 9,61,000. The computationally we could be able to prioritize four molecules. So we bought these four molecules and done animal model testing actually. Seeing this protein folding and folding is an important concept, as all of you know, like. So I'm not going into details of this one. So what I want to emphasize is that the computational biology do play a very important role in varieties of biotherapeutics actually. So here this is a predictor structure of this particular druggable proteins, and we identified a natural product that recognize the protein residues. And here we have tested in the animal model like paralyzed EAE animal model. This is the EAE animal model, which is a paralyzed animal model. And after treatment with the natural product with the small molecule, which we identified computationally, the animal started running actually. I have a video of this one, but due to time constraint, and here you can get also online, the video is available along with that paper which is published also in the scientific reports, and with my colleague actually Krishna Menon actually. So the particular pathway actually how this particular drug has been derived actually, because this will affect both the gut and the brain. So that is the beauty of this particular work actually, gut brain axis actually. So the significance of natural product leading to modulation of this GMF beta in the CNS and gut towards EAE. This is experimental animal model suppression in mice for multiple sclerosis. So this is a pathway actually, or the wonderful signaling mechanism from the gut to the brain actually, like the astrocytes do have this particular um, druggable target, as well as in the gut also in the lumen submucosal flux. And this also intraglial cells also have this particular target drug, where our natural product small molecule will go and bind actually and perform its execute its inhibitory mechanisms I'm not going into the molecular mechanism, but this is also a small molecule derived, which we have got US patent also and published in scientific reports. So it will generally target this gamma beta, and further it will cause the EA suppression in the particular things and pro-inflammatory response and T-regulatory and GM-CSF and brain inflammation get modulated actually, or gut inflammation modulation. So all these things are affected actually brain and gut brain axis. So that is the importance of this particular work, in which both the gut brain axis has been targeted, and we have been able to identify a small natural product compounds which will be able to now planning to do some clinical trial for these particular compounds. And with our Finland collaborators, and here in Namrata also being shown here. I'm not going into details actually, all these things. So finally, I just want to acknowledge like our director, Amrita School of Nanoscience, for allowing me to work in different areas and by collaborating with the different faculties here, like Kushmar Menon and Raja Biswas for microbiological collaboration TB. So all this work has been performed. And the past PhD students and MTech Dr. Jane Jose, she is now in Europe actually, and Dr. Sunita in US actually. So both of them has worked in neurological disorders in the gene of beta, which we identified a natural product. And Dr. Om Prakash, who is a Marie Curie fellowship, and he also completed his doctorate. And Anju CP worked in the MTB area, and she is now in Dubai as a scientist. So all these bright students have contributed the case studies which I have shown here actually, and the importance of the natural product, I think, is a very important topic in the computational biology area, and one has to further deal and exploit more in this particular area. Okay, thank you for your listening.
So thank you so much, sir. This was a very knowledgeable session. So it's time to take some questions now. If you have any questions, please post them. So the first question is how Ayurvedic can be used for personalized medicine.
Yeah, of course, this is an Ayurvedic physician should answer actually. I'm not an IM, but this question is very generalized. Actually, Ayurvedic drugs or whatever we are telling is itself is a personalized only, because the Ayurvedic doctors, by seeing the the Vata, Kapha, Pitta actually, so particular patients those levels will be seen by the doctors. So actually, it is itself is a personalized kind of medicines, and each individual, the Ayurvedic doctors will give personalized kind of medicines only. So it's but in the natural product, it is still a challenge. Actually, we have not yet developed or gone at the natural product level. But whatever the Ayurvedic, like our kashayam and other Ayurvedic drugs, which generally we will call it as a polypharmacology or multiple natural products. So it can the Ayurvedic physicians generally give personalized kind of person to person will vary actually. So they themselves will formulate their own Ayurvedic formulations with respect to the person to person. So due to the from their experience, but computational biology has not yet developed to answer to what extent they are giving to that particular personalized medicines. So there is some limitation to our knowledge at the computational level as well as our modern medicines.
Okay, sir. The next question is technique to select natural products for specific target.
Yeah.
I think she means, yeah, what is the technique to select natural products for specific targets?
Yeah, so these techniques, the in silico techniques you are telling, so in silico techniques can be utilized like molecular docking. One can use for selecting different targets, which target it will go and bind with respect to the docking score. Molecular docking can be identified. Like targets like kinase have different isoforms. So which natural product, which isoform it will go and bind actually select a particular natural product in order to screen with respect to a particular target. Like the target has specific residues, active site residues. So on that basis, also you can create a pharmacophore model, and using natural product derived database, you can screen and get some hits actually with respect to the particular target of interest. So this is a theoretical technique which I feel now machine learning and AI kind of things are also playing an important role, and you can develop some QSAR modeling and can predict actually with respect to some target, if you know, like ACHE or acetylcholinesterase enzymes, where there are a lot of molecules are available, or cancer drugs where you already know the particular target of interest. So on that basis also you can do some QSR modeling also, and can utilize that further for docking purpose, and can identify which molecule is better actually in this natural product derived compounds and its derivatives. So one has to do a lot of exercise before coming into conclusions.
Yes, sir. The next question is, does diosmin have any side effects during the long term?
Yeah, not strictly actually, because it is used as a nutraceutical, and it will generally convert to diosmethin actually, diosmethin. So it is a nutraceutical, so there is no ADMT doesn't make any problem actually. So it can be utilized as a supplement actually. It is already available in citrus fruits, and it is natural product, which is very safe actually.
Okay, sir. The next question is, can repurposed drugs be patented?
Yeah, patent, they may ask you questions like. Mm, you have to, I think, show the formulation aspects or other things and its efficacy. They may ask actually, and it can be a natural product is known actually. So whether it will be patentable is another question actually. But if you formulate that with some other drug like amoxicillin clavulanic acid with diosmin, if I get a formulated drugs, so that can be patentable because we are changing the as such the repurposed one to a formulation, so that can be patentable. And even we are going to get Indian patent for this biosmin also, I think. So it is viable, only you have to show the novelty of the techniques, and how it is being utilized, and what is the prior art, and how whether the patent is really useful actually. So all this thing has to be justified with the attorney and those who are examiners with respect to patent filing.
So I think we are done with all of our questions. So thank you so much, sir. This
Thank you,
was a
thank
very
you.
informative session. So it's now time to felicitate our speaker of the day with a certificate of appreciation. So thank you so much once again, sir, for giving your time. And it was a very great interaction. It was really a pleasure to have you with us here today.
Thanks a lot for inviting me. Thank you. Thanks a lot.
And I'd also like to thank all of the participants for joining. So, if you want more such informative sessions, please stay tuned with us. Please subscribe, like, and follow us for more upcoming events. So, thank you so much, everyone. Thank you so much, sir.
Okay, can I leave now? Okay, thank you. Thank
Yes,
you.
sir.
Okay, thank
Thank you,
you.
sir.